Hello everyone and welcome to today's session. And today we'll be discussing the NCERT exercise for the chapter Principle of Inheritance and Variation. Okay, so this is the part 1 of the lecture. In this session, we'll be discussing some of the questions and the rest of the question we'll be discussing in the next session. So before starting the lecture, I just want to give you one information that the images, text and other materials used in our presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. So what are the objectives of today's session? So we will explore and we will understand the basic concepts, the fundamental concepts of the chapter Principle of Inheritance and Variation. This is the chapter 4 of the NCERT textbook. Okay, by working through the NCERT exercises. So these exercises, they are based on some concepts. We will understand these concepts by discussing these NCERT exercises. So I'll be telling you that on which concept the questions are based. Clear? So in this session, we'll be discussing question number 1 to question number 8. So in total, there are 16 questions. In this session, we'll be discussing half of the questions from question number 1 to question number 8. So these questions are basically short answer type. Okay, very short answer type and long answer type. So there are no MCQs here in the exercises. Now let's begin with this session. So let's read the first question here. So what is the first question? You have to mention the advantages of selecting pea plant for experiment by Mendel. So Mendel is the father of genetics. He gave the law of inheritance and Mendel worked on the pea plant. Now why Mendel selected pea plants? This is the question here. What were the advantages of selecting the garden pea or the pea plant? Okay, you have to answer this. So for more information, you can read NCERT section 4.1. So what will be the answer for this question? So let's have a look at the answer for this question now. So the advantages of using pea plant by Mendel were. So Mendel selected pea plants to carry out his study on the inheritance of characters from parents to the offsprings. Clear? So Mendel was studying how the characters are being transferred from parents to the offsprings. How the traits are being transferred from parent to the offspring. So he was studying the inheritance. Okay, so he selected a pea plant because of the following features of the pea plant. So first one is well-defined characters. So Mendel selected contrasting characters. So what do you mean by contrasting characters? Okay, means if you take height as a character, the plants were either tall or they were dwarfed. So this is known as contrasting characters. There is no intermediate phenotype here. Okay. So peas plant, they have many visible contrasting characters. So you can observe these contrasting characters. So Mendel chose 7 pair of contrasting characters. Okay, one of them was height. The plants were either tall or they were dwarfed. So this was one of the main reason for selecting the pea plants. Then the characters were tall or dwarf plants, round or wrinkled seeds. So this is also a contrasting character. Seeds were either round or they were wrinkled, and you can easily observe it also. Then the pod color. So pod were either green in color or yellow in color. Now flowers were either purple or they were white. So these are two contrasting traits, two contrasting characters. Now next reason, bisexual flowers. So pea plant, flowers of the pea plant, they are bisexual. Now what do you mean by bisexual? Bisexual means they consist of both male and the female reproductive organs. Clear? What is the male reproductive organ? These are the stamens, which consist of filament and the anther, and within the anther, pollen grains are being produced. Female reproductive system is a carpel, which consists of ovary, style, and stigma. So, if the flower is bisexual, then there are very high chances of self-pollination. So, what Mendel did? Mendel crossed the two parents. Then F1 progeny was obtained through crossing these parents. Then this F1 was selfed. So selfing of the F1 gave you F2 generation. Basically, selfing means what? Selfing means self-pollination. So selfing of the F1 means he carried out self-pollination. So F1 plants were reproduced with themselves only. What is self-pollination? When pollen from the anthers, they are transferred to the stigma of the same flower. This is known as self-pollination. This is known as selfing. So this was another reason Mendel chose pea plants, because their flowers are bisexual. Okay, so pea plant, they have bisexual flowers and therefore they can undergo self-pollination easily. Okay, or they can undergo selfing easily. Now, thus pea plant produce offerings with same trait generations after generations. So with the help of selfing, the offerings which are produced, they will have same traits which were present in the parents. In cross-pollination, what happens? There is mixing of the genetic makeup of the two parents. In cross-pollination, what is happening? Pollen from the anther of one flower is being transferred to the stigma of the another flower. Clear? This happens in cross-pollination. So this can lead to generation of some new combination of traits in the progeny. And the traits which are present in the parents, they might be lost. New combination of traits can arise in the offerings. But with the help of self-pollination, the traits can be maintained generation after generation. Now next point, this is very important, cross-pollination. So Mendel carried out artificial cross-pollination in the pea plant. So now a question arises here. Pea plants, they were bisexual. So how did he did cross-pollination? So F1 generation was selfed, but the parents were cross-pollinated. Remember this, parents were cross-pollinated and the F1 generation was self with the another F1 parent, okay, or F1 plant. Now, you can see in pea plant, cross-pollination can be easily achieved by emasculation. So Mendel selected two parents initially. Clear? Both the parents will carry bisexual flowers. Now what Mendel did? Mendel emasculated one of the flower. Emasculation means removal of the anther. So when the anthers are removed, then that flower becomes female flower because the male reproductive system has been removed. Clear? Then the pollen from another flower can be dusted on the stigma of this flower which has been emasculated. This is how Mendel did. Mendel carried out artificial cross-pollination by emasculation of the flowers. So after emasculation, that flower will become the female flower, and then pollen from another flower can be dusted on this flower. stigma of this flower which has been emasculated. And after that, F1 progeny was obtained. And then F1 progeny was selfed to obtain the F2 progeny. Clear? So emasculation means removal of the stamens of the flower without affecting the pistil. So it was very easy to carry out cross-pollination in the pea plants. And why emasculation is being done? To prevent the self-pollination. Mendel wanted to carry out cross-pollination. That's why he did the emasculation of the flowers. Now a pea plant, it has a very short life cycle. The pea plant have a very short lifespan and they produce many seeds in one generation. So if many seeds are produced, if many offerings are produced, then you can study, you can say the results are quite robust. You can study a lot of variations in the offerings. If many offerings are produced, you can study the offerings. There can be a lot of variations in the offerings. So you can easily study the inheritance. Okay, large number of offerings are produced. The pea plant produced many seeds in one generation. And this enables robust statistical analysis. So for statistical analysis, large population, a large sample size should be there. So Mendel, what Mendel did? Mendel was, or he pioneered, he was one of the first scientists to use mathematical and statistical tools to analyze his results. So this enables robust statistical analysis, large number of sample size or large number of offerings. So they were the main reasons why Mendel selected pea plant in his experiment. The next question here, you have to differentiate between the following. So first one is dominant and recessive. So for more information, you can read NCERT section 4.2. Then you have to differentiate between homozygous and heterozygous. And then you have to differentiate between monohybrid and dihybrid cross. So what will be the answer for this question? So see, let's talk about dominance and recessive first. Okay, this is a table showing you difference between dominance and recessive. Now a dominant factor or allele expresses itself in the presence or absence of another allele, which is the recessive allele. So dominant allele or dominant factor, it will mask the expression of the other allele. It's clear? So it expresses itself in the presence or absence of another allele, whether it is present or absent. Okay, then also this dominant factor will be expressed. Now, what are the examples? For example, dominant character in pea plant are tall plant. This is the dominant trait, tallness. And round seeds, this is also a dominant character. Violet flowers, this is also a dominant, you can say phenotype or trait. These are controlled by dominant alleles, which are written in capital letters. So dominant gene or dominant allele, it is always written in capital letter. And let's talk about recessive. Recessive trait is able to express itself only in the absence of a dominant trait or when two copies of it are present. So recessive traits, they are only expressed when the dominant trait or dominant allele or dominant gene is absent. And they are only expressed when two copies of that recessive allele is present. Okay, so recessive traits, they are only observed in homozygous condition. Like, for example, for tallness, there are two alleles, capital T, small t. Capital T is dominant, small t is recessive. Recessive alleles, they are written in lowercase. Okay. So, when small t is present, then only the plant will be dwarfed. Means the recessive trait is only observed in the homozygous recessive condition. when both the recessive alleles are present together. Okay. Example is recessive trait in pea plant, a dwarf plant, wrinkle seed, and white flowers. These are all recessive traits which are controlled by recessive alleles. Now let's talk about next one. Next, homozygous and heterozygous. What is the difference between homozygous and heterozygous? So homozygous, it contains identical pair of alleles for a particular trait. Heterozygous, it contains dissimilar alleles, or it contains two different alleles. Means a contrasting pair of alleles are present for a particular trait. Now let's talk about homozygous. Genotype for homozygous possesses either dominant or recessive, but never both the alleles. So for homozygous individual, there can be two kinds of homozygous individual: homozygous dominant and homozygous recessive. In homozygous dominant, both the dominant alleles are present together. In homozygous recessive, both the recessive alleles are present together. Whereas in heterozygous condition, genotype for heterozygous possess one dominant and one recessive allele. Okay. Then for example, homozygous dominant is capital R, capital R. This is homozygous dominant. Small r, this is homozygous recessive. Then for heterozygous, example is capital R, small r. Here capital R is dominant over small r. And the dominant trait is observed in the heterozygous condition. Clear? So suppose capital R is for round seed, small r is for wrinkle seed. So here the phenotype in the heterozygous capital R, small r will be round. These seeds will be round because capital R is dominant over small r. Clear? These homozygous individuals, they produce only one kind of gametes. Means they are homogametic. They produce only one kind of gametes. Heterozygous, they produce two different kind of gametes. Means they are heterogametic, producing two kinds of gametes. So suppose you take, for example, capital R, small r. This is homozygous dominant. Capital R, small r individual, it will produce all the gametes, and all the gametes will contain capital R only. The small r individual, okay, the person having this genotype small r, it will produce gametes which will have only small r. Whereas in case of heterozygous capital R, small r, half of the individuals will have, half of the gametes will carry capital R, half of the gamete will carry small r, according to law of segregation. Okay, what is law of segregation? Means during the meiosis or during gamete formation, half of the gamete receives one of the allele and the rest of the gamete will receive another allele. So gametes receive only one of the allele. This is law of segregation. Okay, so this was the difference between homozygous and heterozygous. Now let's talk about differences between monohybrid and dihybrid crosses. So here you can see this is the table showing a difference between monohybrid and dihybrid cross. So monohybrid cross is, what is the definition? A cross between organisms differing in one pair of contrasting traits. Clear? Here the parents which are being crossed, these differ in only one trait. Okay, and this trait is controlled by only one gene. Whereas dihybrid cross, this is a cross between individuals which differ in two traits. And these two traits are controlled by two different genes. See here, or you can see a cross between organisms differing in two pairs of contrasting traits. Monohybrid cross example, cross between tall and dwarf pea plant. Okay, this is a monohybrid cross. These two individuals which are being crossed, they differ in only one trait. Okay. You can also write a capital T, capital T. This is being crossed with small t, small t. A dihybrid cross, this is a cross between yellow wrinkled and green round pea seeds. Okay. So here you can see in the case of monohybrid cross, only one trait, inheritance of only one trait is being studied. That is height. Tall and dwarf. This is the height of the plant. In case of dihybrid cross, you are studying the inheritance of two genes. So here the plants which are being crossed, or the organisms which are being crossed, they differ in two traits. Okay. Yellow color seeds and wrinkle shaped seeds. So we have two traits here. Color of the seeds and shape of the seeds. Color can be yellow seeds or green seeds. Shape can be wrinkle seed or round seeds. Clear? Then in monohybrid cross, only single trait is being studied, for example, height. In dihybrid crosses, two traits are being studied, for example, color and texture. Now gametes, monohybrid cross, in monohybrid crosses, two types of gametes are being produced. So in this case, okay, we are crossing two individuals which are heterozygous. So this one parent will produce two kinds of gametes here. Half of the gamete will carry capital T, half of the gamete will carry small t. Okay, this is clear. If there was a cross between homozygous dominant and homozygous recessive, then only one gamete will be produced by each parent. Then, in case of dihybrid crosses, you can see here four types of gametes will be produced. In this case, we are crossing two individuals which are heterozygous for both the traits. So each parent will produce four kind of gametes. So there is a formula for calculating the gamete, 2 raised to power n, where n is the number of the heterozygous traits which are present. So here you can see two heterozygous pairs are present. So 2 raised to power, it will be, you can see 2 here. Clear? Now phenotypic ratio in monohybrid cross, so in the F2 generation phenotypic ratio is 3:1. In dihybrid cross, the phenotypic ratio in F2 generation is 9:3:3:1, which is due to law of independent assortment. Then genotypic ratio in monohybrid cross, this is 1:2:1. Genotypic ratio in dihybrid cross is 1:2:1, 2:4:2, 1:2:1. Means 9 genotypes are being formed here. 9 different genotypes. These are 9 different genotypes. In case of monohybrid cross, 3 different genotypes are formed. Okay, so you should remember the phenotypic and the genotypic ratio in the monohybrid and dihybrid cross. You can get question from here. Then monohybrid cross, this demonstrate law of segregation. So you can demonstrate the law of segregation using monohybrid cross. And you can demonstrate the law of independent assortment with the help of a dihybrid cross. So again, this is very important. So they can ask you in your exam that, okay, law of independent assortment can be explained by monohybrid cross or dihybrid cross. So answer will be dihybrid cross. This ratio 9:3:3:1. Okay, and the genotypic ratio, this is observed due to law of independent assortment. Now let's discuss next question. Question is a diploid organism. Organism is diploid. It has 2n number of chromosomes. This is heterozygous for 4 loci. Okay, how many type of gametes can be produced here? So you can simply apply the formula 2 raised to power n here. So what will be the answer for this question? If you simply apply 2 raised to power n here, n is the number of heterozygous pairs which are present here. So the organism is heterozygous for 4 loci. Locus is the position on the chromosome. So what will be the answer for this question? Let's have a look at the answer now. Now, locus, plural is loci. Now, what is locus? It is a fixed position on a chromosome which is occupied by a single or more genes. So this locus is a position where the gene is present. This is the position on the chromosome where the allele is present or the gene is present. Now, heterozygous organisms contain different alleles for an allelic pair. So, heterozygous organisms, they consist of dissimilar alleles. For example, capital R and small r, these are two dissimilar alleles. Therefore, a diploid organism, which is heterozygous for 4 loci, will have 4 different contrasting characters at 4 different loci. Clear? So, what does this mean? So this means that, let's take an example to understand this. So if an organism is heterozygous for 4 loci with 4 characters, say capital A small a, capital B small b, capital C small c, and capital D small d. So you can see there are 4 genes involved here. There are 4 locus involved here. 4 traits are involved here because these genes are controlling 4 different traits. They are present on 4 loci. Okay, and they are heterozygous, so we will write capital A and small a, capital B and small b, capital C small c, and capital D small d. If in question they were asking homozygous for 4 loci, then we will write capital A, capital A, capital B, capital C, and capital D, capital D. Okay, so this is how you represent organism which is heterozygous for 4 loci with 4 characters. If they were saying 5 loci with 5 characters, then you can add E, capital E and small d here. Now what happens during meiosis? It will form 16 separate gametes here. So then during meiosis, it will segregate to form 16 separate gametes. So how 16 gametes will be formed? Clear? Now the number of different type of gametes in a heterozygous individual can be produced. This can be determined by the formula 2 raised to power n. So you have to use this formula 2 raised to power n. What is n here? N is the number of heterozygous gene pairs. So how many heterozygous gene pairs are present here? So 1, 2, 3 and 4. So there are 4 heterozygous gene pairs here. So you can replace this n with 4 and you will get the answer 16 then. So therefore the number of gametes produced will be 2 raised to power 4 or 16, as the organism has 4 heterozygous gene pair. If there are 5 heterozygous gene pair, so you can write instead of 4 you will write 5. If they have 3 heterozygous gene pair, instead of 4 you will write 3. If they have 2 heterozygous gene pair, you will write 2. This is how you calculate the number of the gametes. So this is very important. You should remember this formula. So you can see here, this is a diploid organism which is heterozygous for the, which is heterozygous for 4 loci. This locus is the position on the chromosome where the gene is present or where the allele is present. Okay, so they are showing you this homologous chromosome. On one chromosome, capital A is present, and on the other chromosome, at the same position, small a is present. Then capital B is present. Then on the another chromosome, okay, homologous chromosome, capital B is present at the same position. So this is the genotype here: capital A, small a, capital B, small, capital C, small d, capital D, small d, and the number of gametes produced will be 16 here. The next question, you have to explain the law of dominance using a monohybrid cross. So monohybrid cross is a cross between individuals which are differing in only one trait. So what will be the answer here? How can you explain law of dominance using a monohybrid cross? So let's have a look at the answer. The law of dominance. So law of dominance says that one of the allele masks the expression of the another allele. So dominant allele expresses itself in a monohybrid cross and suppresses the expression of the recessive allele. So dominant allele suppresses the expression of the recessive allele. However, this recessive allele for a character is not lost and remains hidden or masked in the progenies of the F1 generation. So what happens in the F1 generation? Only dominant trait is being observed. Only one of the parental trait is observed in the F1 generation. And the recessive trait is not observed in the F1 generation. Okay, why the recessive trait is not observed in F1? Because of the law of dominance. So in heterozygous conditions, so F1 hybrid, they are heterozygous. In heterozygous condition, one of the allele is dominant over the other. That's why only dominant trait is being observed. One of the parental trait is being observed. Clear? And the recessive trait is not observed. The F1 generation. So these recessive traits then reappear in the next generation. So when selfing of the F1 is done, then this trait reappears in the F2 generation. That is mainly because of the law of independent assortment. Let's have a look at the example now. Okay, let's understand dominance with the help of example of a monohybrid cross. So when the pea plants with round seeds having the genotype capital R, this is crossed with the plant with wrinkled seeds having the genotype small r. All seeds in the F1 generation were found to be round. So round is a dominant phenotype here because in F1 only round seeds are observed. Okay, and the genotype of the F1 will be capital R and small r. Now when these round seeds were then self-fertilized, when the selfing of the F1 generation was done, both the round and the wrinkled seed appeared in the F2 generation in 3:1 ratio. Okay. Now this 3:1 ratio can be also explained with the help of law of dominance. Because 3 was dominant, 1 was recessive. Okay. This law of independent assortment is valid in the dihybrid crosses. Now, hence in F1 generation, the dominant character round seeds appeared and the recessive character wrinkled seed got suppressed. So round is a dominant trait which is controlled by capital R. That's okay. And this recessive trait, this reappeared in the F2 generation. So here you can see these are the parents. Parents are what? Having round seeds. One of the parent is having round seeds. Another parent is having wrinkled seeds. Genotype is also given capital R and small r. So they are homozygous for a particular trait. Okay, both the parents are homozygous. Then you can write the gametes which are being produced here. The gametes produced by the parent 1 having capital R. It will produce all the gametes having capital R. Then the other parent having wrinkled seeds, it will produce gametes having small r only. So in the F1 generation, after fertilization, capital R and small r is being produced. So all the plants will have capital R and small r. Okay. This is clear, means the plants will be having round seeds. All the plants will be having round seeds. Capital R is dominant over small r. Capital R is masking the expression of the small r. Small r is for the wrinkled seeds. Now selfing of the F1 generation is being done here and you get the F2 generation. In the F2 generation, you can see you are getting the phenotypic ratio 3:1. 3 dominant and 1 recessive. Okay. Clear? Here the genotype is also mentioned of the F2 generation. Okay, capital R, this will be round seeds. Capital R, small r, this will be show the phenotype round seeds. Capital R, small r, this will also show the phenotype round seed, and small r, this will show the phenotype wrinkled seeds. Here you can see this recessive trait only appears in the homozygous condition. Okay, so this is a figure showing a monohybrid cross between round and the wrinkled pea plants. The next question, you have to define and design a test cross Question is regarding test cross. So what is a test cross? So let's have a look at the answer. So test cross is a cross between an organism with unknown genotype and a recessive parent. So test cross is done. What is the purpose of the test cross? Test cross is done to know the genotype of the parent showing dominant phenotype. Okay, I am repeating this again. Test cross is done to know the genotype of the parent having dominant phenotype. And how this is done? The organism having the unknown genotype. Okay, we want to know the genotype of this organism which is showing dominant phenotype. This is being crossed with the recessive parent. Okay, now there can be two cases here. Clear? So what is the purpose of test cross? It is used to determine the individual's genotype. It helps in determining if the individual is homozygous or heterozygous for a trait. So now you know that in the F1 generation, okay, the plants were heterozygous, but they were showing the dominant trait. So by observing a plant, by observing any individual, you cannot tell about their genotype. You can only tell about their phenotype. Okay, for example, you can say plants are tall, but you cannot say the genotype is capital T or capital T small t. So you can know this by using test cross. Okay. Now, if the progeny produces all the dominant traits after this cross, okay, if the progeny is producing all the dominant traits, then the unknown individual is homozygous for that trait. So let's understand this using this image here. So you can see in first case, if the unknown genotype, if the organism having unknown genotype, if that organism is homozygous for that trait, then what will happen in this cross? This is being crossed with a homozygous recessive parent. Now, gametes produced by this parent will be, all the gametes will carry capital R. Here, the homozygous recessive parent, all the gametes will contain small r. Then you can see only the dominant phenotype is being observed here. All the plants will having capital R and small r. So suppose capital R is for round seed, small r is for wrinkle seed. So all the plants will having round seeds here in 1:1:1:1 ratio. So in test cross, remember you get 1:1 ratio only. And if in the offerings, if in the progeny you are getting only the dominant trait, then what will happen? Means that parent will have homozygous. That parent is homozygous for that trait. Now let's take another case. So in another case, what is happening here? You can see if the parent is having heterozygous genotype, capital R and small r, it is being crossed by homozygous recessive parent. Then if the F1 generation, if in the F1 generation or in the progenies, 50% dominant trait is observed and 50% recessive trait is observed in 1:1 ratio, then you will say that individual will be having heterozygous genotype. Okay. So here you can see this heterozygous individual, this will produce two kinds of gamete. Half of the gamete will carry capital R, half of the gamete will carry small r. Homozygous recessive parent will produce all the gametes having small r. Then in the F1 generation, in the progeny, you will get 1:1 ratio of capital R small r and small r small r. So if you are getting both the traits in 1:1 ratio, okay, if you are getting the dominant phenotype also, 50% are dominant and 50% are recessive. 50% of them are showing recessive phenotype. Then you will say that the organism having dominant phenotype, that is heterozygous for that character. Okay, so remember this is very important test cross. You should know the purpose of the test cross and you should know both of these cases. The next question, using a Punnett square, work out the distribution of phenotypic features in the first filial generation, means in the F1 generation. First filial generation means first generation. After a cross between homozygous female and a heterozygous male for a single locus. So the female is homozygous. Okay, male is heterozygous for a single locus. Okay, means it is a monohybrid cross. So what will be the correct answer here? Let's have a look at the answer. So you can see, let's take an example of guinea pig now. Okay, in guinea pig, suppose males are heterozygous. Okay, for the black color coat. Heterozygous means they will have dissimilar alleles, capital B and small b. This heterozygous male is crossed with the female, which is homozygous. Okay, so she is carrying small b, small b. So capital B is for black coat color, small b is for white coat color. And capital B is dominant over small b. So basically you have to cross capital B small b with small b small b. So the male will produce two types of gametes here, capital B and small b. Males are heterogametic, they are producing two kinds of gametes. Okay, where the female will produce only one kind of gametes, all the gametes will be having small r. All the eggs will be having small r. All the sperms will be having half of the sperms will having capital B, half of the sperms will be having small b. Now, how you can calculate the genotypic and phenotypic ratios of the F1 now? Okay, you can make a Punnett square and then you can calculate the genotypic and the phenotypic ratio here. So you can see the parents are having capital B, small b and small b, small b. Capital B, small b, these are heterozygous. Small b, these are homozygous. Okay, gametes, you can write the gametes which are being produced here. Okay, this heterozygous individual is producing two kinds of gametes. Homozygous will produce only one kind of gametes. Then you can see, you can make a Punnett square. On one side you will write the gametes of the female, one side you will write the gametes of the male. Then you can make all the possible combinations and you can calculate the genotypic and the phenotypic ratio here. So here, you are getting two black coat and two white coat. Okay, this is 2:2, or you can write this as 1:1. Okay, so genotypic and phenotypic ratio in the F1 generation is 1:1 here. Now, next question, when a cross is made between tall plant and yellow seeds, the genotype is given to you. They are heterozygous for both the traits. Okay, this is crossed with tall plants with green seeds. This, okay. Now, what proportion of phenotype in the offerings could be expected to be tall and green and dwarfed and green? So what proportion they are asking of the phenotype in the offerings could be tall and green and dwarf and green? So what will be the answer for this question? Now how to calculate this? So what is given? Genotype of the parents are given to you. You have to just make a cross. First you have to make the gametes. You can then make a Punnett square. Then you can calculate the progeny. Okay, the probability. Okay, probability is in the progeny. Now a cross between tall plant and yellow seeds and tall plant and green seeds is being carried out here. Capital T is tall for tall, and this is the dominant allele. Small t is for dwarfness, and it is recessive allele. Capital Y is for yellow and dominant. Small y is for green color seeds, and this is the recessive allele. So first of all, this is a table showing you parents' phenotype, genotype, and the gametes produced by the parents. Okay, so the phenotype of the parents are tall. One of the parent is tall with having yellow seeds. Another parent is tall and is having green seeds. Genotype is also mentioned here. This is given to you. Genotype of one of the parent having tall and yellow seeds will be capital T, small t, capital Y, small y. And then for the tall and green seeds, this parent is having the genotype capital T, small t, and small y, small y. Then the gametes which will be produced here, again you can calculate the gametes here using the formula 2 raised to power n, and then you can write the gametes also. So this parent having the phenotype tall and yellow seeds, this will produce 4 kind of gametes. Then another parent will form 2 types of gametes only. Now, okay, again, we know about it. Now, let's calculate the genotype and phenotype of the offerings now. Okay, so you can make a Punnett square. We have calculated the gametes which were formed here by the parents. So you will write gametes of one of the parent on one side, gametes of another parent on another side. Then you can make all the possible combinations using the Punnett square. Clear? So this is the Punnett square. You have written one of the gametes of one of the parent on one side. Then you can write gametes of the another parent on another side. Then you can make all the different combinations here. So all the genotype is also mentioned and the phenotype is also mentioned here. So after analyzing this table, what you can see here. Okay. So how you make different combination? This goes with this goes with this. Okay, then this goes with this, and then this one goes with this. Similarly for the other gamete here. Okay. Now there will be 3 tall with green seeds. So they were asking the question tall and green. Okay, tall plants with green seeds. So there are 3 tall plants having green seeds. Genotypes are capital T, small y, small y. These are tall plants having green seeds. This is also another capital T, small y, small y. And then capital T, small y, small y. Okay, then next question was dwarf plants having green seeds. So one dwarf plant with green seeds, genotype will be small t. So this is here. You are getting only one genotype here. Okay, small t, small y, small y. This will be dwarf plant having green seeds. So this is homozygous recessive for both the traits. Clear? Now what is the next question here? 2 heterozygous parents are crossed. If the 2 loci are linked, what would be the distribution of the phenotypic features in the F1 generation for a dihybrid cross? So they're talking about dihybrid cross means it is involving 2 traits, 2 genes here. But they are saying the 2 loci are linked together. Then what would be the distribution of the phenotypic features in the F1 generations? So what would be the answer for these questions? Well, let's have a look at the answer now. So let's define linkage first. So linkage is defined as the coexistence of 2 or more genes in the same chromosome. So what happens when the genes are linked or when linkage occurs? So for linkage, the 2 genes should be present on the same chromosome, and these 2 genes should be present very near to each other. So the distance between 2 genes should be very less. They should be present on the same chromosomes. And because they are linked, they are physically associated with each other. So they are inherited together in the offerings. And when genes are linked, then more parental combinations are being produced as compared to the recombinants. Okay. So what would be the distribution of the phenotypic features? Here you will get more parental combinations as compared to recombinants because the genes are linked. So linked genes are situated on the same chromosome and they lie close to each other. They are inherited together and are said to be linked genes. So they are inherited together. That's why more parental combinations are seen. More parental types are seen as compared to non-parental. As the distance between the 2 genes is very less, the recombination between the two genes will be less. Okay. By recombination, new combinations are being produced. So here there is no recombination because of linkage of the genes. So parental combinations are more. Okay, let's have a look at the example. So there is a cross between yellow-bodied and white-eyed Drosophila. Okay, with wild-type parents. So for yellow-bodied, you have Y gene. For yellow eye, you have W gene. Okay, so you are crossing this Drosophila with wild-type Drosophila. Wild-type Drosophila, the genes are represented by Y plus and W plus. So this plus in the superscript, this represents the wild-type genes in the Drosophila. Okay. Okay. Now, so this cross will produce wild type and yellow-white progenies. Clear? It is because yellow-bodied and white-eye genes are linked together. Therefore, they are inherited together in the progenies. So you can see, let's understand this with the help of this image. This is Y and W here. Okay, Y and W. Clear? So there is one female Drosophila which is being crossed by male Drosophila. So male have X and Y chromosome. Females have X and X chromosome in case of Drosophila. Okay, now you can see distance between Y and W is very less. By observing this, you can tell that the genes are linked. Y and W are distance between Y and W is very less. Okay, so Y and W genes, they are linked together. So if they are writing yellow-bodied and white-eyed genes, they are linked together. Okay, so these are the two parents. You can make the gametes. Then you can calculate the F1 generation. Then after F1 generation, you will get the F2 generation. So you can see parental combinations are more here as compared to recombinant. Parental combinations are 98.7%. Recombinants are only 1.3%. So you are getting more parental combinations. So whenever you get more parental combinations, means the two genes are linked together. Okay, so Y and W genes, they are linked together. So you are getting more parental combinations here. The recombinants are very less. Non-parental are very less. Okay, so this is happening. Why? Because these two genes are very closely linked. Which two genes are closely linked? Y and W. Okay, these two genes are also very linked. Y plus and W plus. The wild type genes and the other genes. These two are linked together. Okay, this is clear. All right, so we have discussed the exercises. Okay, question number 1 to 8. We'll discuss rest of the exercise in the next session. So we'll end this session now. I hope this session was quite knowledgeable for you. We will meet you in the next session. Thank you very much for listening.